Hello, my name is Bashir. I'm a professor in bioengineering and also the Dean of the Granger College of Engineering at the University of Illinois at Urbana-Champaign. The title of my talk today is Point of Care Sensors for Detection of Disease Biomarkers and how this might apply for predicting pandemics. I want to thank NSF for organizing this workshop and for inviting me to come give a talk. Our group is interested in using micro, macro, and nanofabrication approaches to develop technologies that can be applied to grand challenges in cancer, sepsis, and global health. We're also interested in developing biofabrication and cellular systems for drug screening, biorobotics, and hyperorgans. Today, I will really talk about this right side of the slide of this right quadrant of our work. So I'll give you a case study around sepsis stratification work that's going on in our group and how they might apply to what we're trying to do. Talk about SARS-CoV-2 diagnostics and point-of-care sensors, and finally give some thoughts on pandemics prediction and the work here at UIUC. So let's talk about sepsis first as a test case. Sepsis is actually a critical condition in hospitals. It's defined as a life-threatening organ dysfunction due to dysregulated host response to infection. It's a leading cause of death in critical care in the US and the world. It's a huge cost burden, and it is the most expensive condition and the leading cause of readmission in hospitals. 1 in 3 individuals who die in hospitals have sepsis. And there are more cases of sepsis in the US than prostate cancer, breast cancer, and HIV combined, and also prostate cancer, breast cancer, and lung cancer combined. So it's a big challenge. So just some further information on sepsis. In 2016, a task force was convened by Society of Critical Care Medicine and the European Society of Intensive Care Medicine. And that's where they came up with this updated definition, third definition of sepsis that I mentioned earlier. And what they did was they provided a sort of an approach to quantifying as best as they could this very complicated syndrome. And we came up with this sepsis-related organ failure assessment called SOFA score. And there's 2 types of SOFA scores used today. One is called QSOFA, which is a quick bedside prompt, just looking at 3 parameters here: respiratory rate, mental status, and systolic blood pressure. But then as the situation for the patient perhaps gets worse, then there's a more detailed SOFA score, which has more variables. So the idea is to use these parameters to come up with a score and use that score to decide the course of treatment. We think what's important here is to also develop this predictive capability as best as possible using building a large database. So can we build a large database where we could identify different clusters of people in different stages? When the biological reaction is a pro-inflammatory stage, then there is an anti-inflammatory stage that could then lead to further organ failure or septic shock. So what we did was to also look at what happens in the hospital today, just for essentially our general information. So this is what happens, of course, pre-hospital. You get an infection. You start to feel abnormal. That might take a day or so. And then you might not even go to the ED depending on how you feel. Maybe your immune system will fight the infection and you will be fine in a few days just being at home. But if things don't get any better, then you go to the emergency department. It could take a few hours to get blood culture ordered and also perhaps some prescription of antibiotics. But the blood culture results don't come back for 5 days, for up to 5 days. You need up to 5 days for a negative. It could take another hour for identification and another day for determining which drug to use. And then, of course, if the patient is in the hospital during this time, it could take many days for the recovery or, unfortunately, death. So many challenges here, as you can see. Blood culture takes too long, and also 50% of them come out negative. So physicians they have to make decisions. They can't wait for 5 days. How to prioritize and stratify this antibiotic treatment, and also how to quantitatively profile the patient immune system over time. So we launched a study working with our local hospital, Carl Foundation Hospital, where we pulled an interdisciplinary team together of our clinical partners from the hospital, microbiologists, machine learning and data analytics, experts from our campus, and of course our group, which does the sensing part for a living. And essentially, we set up a study where every patient that gets a blood culture order, we also get the blood from the patient. And we measure these markers that are known in literature to be affecting one of the pathways of sepsis. So these are proteins and cell markers, both pro-inflammatory and anti-inflammatory. And then we also get the entire electronic medical record for that patient stay. We take all their data and we develop some machine learning algorithms to then, the goal is to help us identify which biomarkers are most important. These are all the parameters that were measured on the x-axis, and the y-axis is the normalized feature coefficient, which essentially gives us its importance in the outcome, in the outcome of the patient. And as you can see here, what was also kind of known is that sepsis there's no one biomarker for sepsis. There's multiple parameters, multiple biomarkers that can affect sepsis. And perhaps we need to develop a panel. So for example, IL-6 is strongly correlated to the outcome, PCT, MMP9, and so on. You also have the white blood cell count and also the CD64 count, which is a cell marker. So point is that this is sepsis is a complicated syndrome. You need to measure these multiple markers and then use a composite score from these markers based on this large data set that we collected. Of course, what is also really important is to compare this to what's done today in the clinic. So what you see here is the AUC curve, which kind of tells you the predictive power. And if you look at the QSOFA and SOFA approaches applied to our data set, it's really interesting. This gives you basically an AUC of like 0.5 and 0.4, which is essentially like a coin toss. So today, what physicians are actually doing in the clinic is very qualitative. Right? And now if you start to add these biomarkers to it and also the EMR to it, the predictive power absolutely goes up significantly. This is pretty good already. And our new data set actually shows this to be even higher. What's also important is this plot, which shows that the measurement from a drop of blood at T0 right here actually gives you the same AUC as if you were collecting the electronic medical record data for 16 hours. So you could save 16 hours by doing a measurement at T0 and getting the results from these biomarkers if you apply our approach. So this work is being translated to a company called Prognosis, just for full disclosure. I have an ownership in the company. It's Bobby Reddy. CEO now has taken this in a great direction where they're building a very large data set, a proprietary set of biological and clinical time series data from past patients. Then developing a software, which this advanced machine learning tool to then learn from this past outcome to predict the outcome of the next patient. And then also a point of care device, which could accelerate the analysis of the panel of biomarkers. Okay, let me now switch gear to SARS-CoV-2, which is certainly in all of our minds right now, and to talk about what to test and what are the different tests that you hear about. So first of all, also the sample sites. Where can we collect the sample? We all have taken the dreadful nasopharyngeal swab, which is a swab that is inserted into the nose, deep into the nasal cavity, all the way up to the brain. We have also done work recently with saliva, and that's what we're using on our campus, where we believe saliva can be a really good alternative to the MP swab. And certainly more and more in the clinical space, the nasal swabs are becoming also more popular because they're easy to implement, and you can do this yourself. Now, if you go through some of these drive-thrus, they give you a kit and you do the nasal swab in your interior nostrils yourself, and put it back into the fluid. So those are the two most common sources of samples for detection of SARS-CoV-2. Now, what is tested? So what you can test is the RNA from the virus. You can also look for the proteins from the virus, which are these two proteins called the spike protein and the S protein and the N protein. Okay, and then of course there's also antibodies in the blood, and we look for IgM or IgG, and these antibodies indicate the immunity. So whether you have had been infected and now are immune because of these antibodies, or if you're vaccinated, you have these antibodies that are produced. So there's three types of tests that you hear about. One is the molecular test. Molecular tests are based on looking for the RNA through PCR or through isothermal processes such as RT-LAMP or also RT-LAMP plus CRISPR. These tests are really the gold standard. They are very sensitive, and it's because the target is amplified. You can detect even a few copies of that RNA, and the inherent process of PCR amplifies that target by billions. That test costs about $15 or so. The basic cost for that is about $15, and it could take, the test alone can take between 30 to 90 minutes. And if you take into account the sample collection and also the data transmission back to the patient, this can be days, by the way. So as we know, there's long lines here. This is where the whole, how you do it, how you scale it up becomes really important. And in most big cities, this is taking many days to get results back. You also hear about antigen tests. Antigen tests are inherently less sensitive, about 100 times less sensitive than the molecular tests. Here, the target is not amplified, even though there are now reports in research of folks developing very sensitive antigen tests. But what's available in the market today is actually about 100 times less sensitive. And here you're looking for these proteins, essentially, or fragments of the virus. Think of it that way, right? So you're looking for these pieces of the virus that are connected to the membrane, perhaps the S protein or the N protein, or in some cases, maybe the whole virus might be captured also, but there's no way to know. And these tests are more available at the point of care. They're cheaper, about $3 or so, and they do take much less time. But as you can see here, you are giving up on sensitivity to lower the cost and lower the time. So if someone can develop a molecular test that is actually in a few dollar range and can take 5 minutes or less, then that's kind of really the holy grail, and that can truly displace the antigen test completely. But due to the lack of availability of such a molecular test today, antigen tests are becoming more and more popular. As a matter of fact, just a couple of weeks ago, the Biden administration pushed to work with some companies to scale and bring these tests to the US at much larger scale. Also, you hear about antibody tests. The antibody tests are looking for antibodies, as I mentioned. These have sufficient sensitivity. These tests, target is not amplified. Again, these are available at point of care. They have a very similar format, actually, of testing as the antigen test, and they're very similar in cost and time. Where to test? This is also important as we talk about scaling of testing, right? So the fact is that today, the samples are collected, whether it's through a drive-through, whether you go through a hospital, or now there's also kits available for home for the sample collection. But then the sample is taken to the lab, and the lab is where the sample is tested. And again, there, because of the scale of the lab, you can do thousands of tests per day. You can take part of that lab, the essential elements, and put it in a mobile trailer. And that trailer could do thousands per day. As a matter of fact, I'll give you an example of that later. You can also imagine a form factor that is a benchtop system, which you need a trained health professional to operate. But this could be placed, for example, on a table. It's not something you would move too much, but you could put it in a mall, at an airport, at a stadium, and this could potentially do hundreds per hour. The interesting situation today is that such a solution doesn't exist. There's actually no company making such a product, but there's a very clear need. The business case is perhaps has to be justified. And then finally, potentially a suitcase format, which can do tens per hours. And then eventually the handheld, which is just a few tests per hour, I would say, and a person could do it themselves. So you can see as you go from left to right, the throughput goes down, but the size also goes down, and the portability increases. Okay, so in our group, we have been for many years actually working on viral load on a chip, and we have demonstrated detection of HIV, HCV, Zika, dengue, and chikungunya. This is parts of this work in collaboration with Professor Brian Cunningham and Professor Bill King here at UIUC. And the idea here is to develop a cartridge which can take a drop of blood. The work we had done was for a drop of blood, process that blood on a chip, and then amplify the RNA of these viruses and use a cell phone camera and a small apparatus to detect the amplification. And this, works. The spec needs to be improved clearly, but it is something that we were able to build upon very quickly this year to then develop a test for SARS-CoV-2, a portable test. This is using our RT-LAMP approach. So the sequence for SARS-CoV-2 came out on January 10th. And as soon as the sequence came out, then many groups, including ourselves, looked at that sequence and we developed our own primers, which are the molecules that can recognize specific parts of the sequence to identify that this is SARS-CoV-2. So we developed our own primers and we were able to, this paper was published in actually late summer, early fall, but we were able to get samples from OSF Healthcare for the viral transport media from patients, from swabs, and we used our approach for the RT-LAMP process, and then also used, compared that to the clinical part, the gold standard of the RT-PCR. These tests were actually done in an additively manufactured cartridge, which was developed in collaboration with Professor Bill King. He's a leading expert on digital and additive manufacturing. So you can see here a plastic cartridge that's the size of a credit card. And then this cartridge is then inserted into a reader, which has a cell phone camera to then detect the fluorescence amplification and get you a yes/no signal. And the results look pretty reasonable. This data, they look pretty good. This data here shown the fluorescence intensity that we measure from the cell phone camera as a function of time, the 10 minutes, 20, 30, and 40, and you can see a statistically significant difference in, let's say, 30 minutes between the positives and the negatives, and get very good specificity and sensitivity. So we believe these technologies can be scaled up. Again, they take, in this case, about 45 minutes, 30 to 45 minutes, but they can be done at a point of care setting. We also then further improve this process working with Carl and OSF to then get samples for saliva and VTM. So in this case, we got samples for VTM and saliva from the same patient to then compare these two sources of the sample. And you can see the results here. The basic summary is, the basic message is that from VTM and from saliva, the data looks very comparable. On the y-axis, you see the threshold time, which can be in the 20 to 30 minutes. So the detection time can be in the 20 to 30 minute. And we have an optimized process that can detect even lower copies per microliter, both from saliva and from VTM. In the past, we have also developed actually microfluidic cartridges that can measure cells from blood, or it could certainly be from other samples also, but we have demonstrated this from blood. And this is important because certain types of cells and molecules and proteins, as I mentioned earlier also, they can indicate the status of the immune system, how strong or how weak the immune system is. So this device can actually take whole blood, and this is whole blood cells flowing in a microfluidic cartridge. We can lyse the red blood cells, we can recover the white blood cells, we can electrically measure those cells, capture them on a chip, specific types of cells, and then count again electrically to get the count for the captured cells. And we've demonstrated this for CD4, CD8. These two are markers of HIV, and CD64 is a cell marker for inflammation. And this work has been published. So another thing I wanted to mention that's also important to the topic today is a project that we have ongoing funded by DARPA, and this is in collaboration with Professor Bill King, who is a PI, where the goal is to reduce the time of manufacturing at scale, which today can be up to 5 years, down to 24 hours. So from the time that you have validated prototypes and field tested with some validated chemistry, how quickly can you deploy those tests to a user? So the goal in the DARPA style is we said, how about 24 hours compared to many years? Right? So this project is ongoing, and the goal is to sort of develop the framework and develop the vision of how this would be done. And we believe this can be done, where clearly it takes more than a village. It'll take the world to come together. But there could be, you can imagine, once a test has been uh, validated, then you develop a virtual warehouse for that assay, which has all the manufacturing files, all the details of the assay chemistry, and all of the sort of components, a digital twin of all the components. And then there are digital manufacturing sites that you put all around the world in a network, which would produce on demand the cartridges and the chemicals, the RNA, potentially the proteins, whatever is needed, can then be all produced locally so that you even save time for transport, essentially. And we believe this can be done, but you can see this obviously is a big coordinated effort. So let me, in the last 5 minutes or so, just talk about some definitions and can bring these thoughts together. So we're here obviously to talk about pandemics. WHO defines a pandemic as a worldwide spread of a new disease. CDC defines pandemic, again, as an epidemic that has spread over several countries or continents, usually affecting a large number of people. These are caused by pathogens that can be transmitted via air, water, environmental contact, or body fluid exchange. Animals, insects, humans, or as we know now, crossovers across it through, which is one of the biggest risks. They're caused by viruses, such as SARS, MERS, or all viruses, HIV. There could be bacteria, so plaque, was actually produced by bacteria, or they can be fungi, which are much slow growing actually, such as mold and yeast are a form of fungi. And then there is this very important question about knowns versus unknowns, right? Talk about a new disease here, which means clearly the pathogen is new. So you need to try to predict, right, when and how a new pathogen might come about. And then how do you get a sample of it to get a sequence for it? And then how do you use that to quickly scale up to address the need to help people? So there might have to be a systems engineering approach for this, where we have to develop sort of what I would say pre-indicators of the pandemic. So there's multiple criteria, and perhaps similar to sort of drawing the analogy from sepsis, where there's many markers that might have to be tested as a panel. Perhaps there are many different indicators or pre-indicators in this case: environmental, epidemiological from animals or humans, population and illness data, food, water supply, social media, and perhaps others. So we need to be monitoring this, defining what they are and then monitoring them. What is really important, of course, for these biological pathogens is to collect them. You have to, at the end of the day, get a sample and sequence it. The sequencing technology have advanced tremendously in the last many years, in the last 10 years. And now, given samples that are collected, within a matter of weeks to months, you can get the full sequence. Within a matter of weeks, you can get the full sequence. And perhaps this should be done for all pathogens. So this is clearly a monumental task, but perhaps you can do that on highest risk first. And this is where scientists can inform us about the current ones and which current ones have the highest probability of mutating and causing us problems in the future. Once you have the sequence, then the question is, how quickly can you develop a test, and what to test, where to test, and how often to test, right? But I believe the test can actually be developed relatively quickly. So water, food, environmental samples, human samples, where do you test it? A pandemic clearly, as per definition, is crossing countries and crossing boundaries of continents. So perhaps at airports, train stations, seaports, perhaps there's climate and satellite data and other, considerations around where, what, and how often to measure. And then obviously developing mitigation strategies immediately, right? Travel policies, quarantines, and public health measures, and using obviously this information to refine all these previous steps. I think it's also really important to, in this new age of data science and data and machine learning, one can actually do this in a way where the data is collected, and whether it's structured or unstructured data, but then be able to use that data to predict the future even better using machine learning and AI. Of course, what I didn't mention is that as the sequences are determined, one can clearly launched drug development efforts. And again, there's been some heroic effort to develop the vaccines for SARS-CoV-2 this year in record time. The diagnostic scale-up could have been done a lot better in our country and worldwide, but we believe it can. And hopefully next time we'll be better prepared. And then obviously deployment and distribution of both drugs and diagnostics to the people that need it. I want to take just a couple of minutes to talk about our campus efforts in terms of keeping our communities safe. So our provost charged a team early summer, late spring, early summer. It's called SHIELD, which is the idea that this SHIELD is protecting our campus community. You've probably heard about our testing and this whole program on the news. I like Marty's quote here, who is the SHIELD project leader as professor in chemistry in our new College of Medicine, that failure is not an option. So we developed our own approach on campus to target, which is algorithms for prioritized testing. Then testing itself, we developed a saliva-based test, which we believe is fast and scalable. And then a digital exposure notification in partnership with the public health via the Safer Illinois app. SHIELD is one of the largest mandatory COVID-19 screening efforts at any university in the world. As a matter of fact, during the August-September timeframe past fall, we were performing about 2% of all the COVID-19 tests in the US were happening in Urbana-Champaign. All university faculty, staff, students who come to campus must be tested. Results are provided within a few hours, and access to campus building requires a negative test. So we have a whole system that's working where samples are collected, saliva samples are collected from 19 sites across the campus. They're transported to a central lab. The lab is running 24 hours a day, 3 shifts, and we get results back on the app within a matter of a few hours, 4 to 5 hours, we get results back. We have now taken this campus effort, which is called the COVID-19 SHIELD UIUC effort, and also scaled it across the state through an effort through what's called SHIELD Illinois. And then also we are scaling this nationally through SHIELD T3 Health to developing mobile trailers. So 6 of these trailers are now already at different parts of the country. They can do 5,000 to 7,000 tests per day. And then we are deploying more of these mobile trucks across the country. This is the dashboard as of yesterday. We have tested 1.4 million samples already. This last 7-day case positivity rate is 0.42%, which is the lowest in the state of Illinois. And we've kept that to less than 1% for the last 8 months. There was a problem when students came in last fall, but we immediately put measures in place to control that. And since then, things have been relatively under control. And this is the number of tests that are performed every day. You can see every day, ranges from 5,000 to 6,000 all the way to 15,000 tests per day. So with that, I'll stop. I want to thank all of my collaborators, students, and the clinical partners that helped us do all the work that I presented from my lab. And I want to certainly thank our campus, and I'm so proud of the SHIELD team for everything it has done that many of us have contributed to keep our community safe and healthy. Thank you.